All right, this lecture is going to cover the introduction as well as chapter 1.1, Principles of Science. So when you're done reading this, you should be able to explain that science consists in testing, possibly rejecting, and improving our simplest and best explanations using data, not improving the correctness of a particular conjecture. So again, science is for testing explanations, and it has nothing to do with proving ourselves to be correct, which is not really what we see in modern discourse, but this is sort of the core of what science is. Here's a nice picture of the evolutionary diversification of Hawaiian honey creepers. There's this nice tree here. But we got to talk for just a moment about what zoology is. Zoology is the scientific study of the animals. Who are the animals? Well, the animals are a clade or a group of organisms that fall under the eukaryotes. Eukaryotes is the domain of organisms that animals belong to. We'll maybe talk about that later. But how do we study organisms and their relatedness? Well, we use a phylogenetic tree. You have to be able to use a phylogenetic tree in order to be able to understand how animal life is related. So a phylogenetic tree is a diagram depicting the history of animal life. Branches represent evolutionary lineages, and each branching event represents a historical splitting of an ancestral species to form new ones. So let's take a closer look at our phylogenetic tree that we have up here. Sorry, it's a little blurry, but let's start by thinking about, first of all, what's at the tips here represent different taxa. In this case, they are different tribes of butterflies. And you can see each split here represents a diversification. So everywhere that you have one of these branch points, a split has taken place, and you've gone, you have accumulated different taxa or different tribes. In this case, it could be species, it could be genera, but you should also know that we're working from left to right in this case. Time is working in this direction, so the more ancient species are happening over on the left, more modern species are on the right. And so everywhere where it splits represents a ancestor or a common ancestor from everything downstream of it. So the common ancestor between the Libellithinae and the rest of these groups, their common ancestor is right back here. So right where it splits and goes to these various groups. So let's answer this question really quick. Where is the most recent common ancestor between the Callinicrae and the Charaxidae? Okay, so here is our, Callinicrae and Charaxinae. Where is the most recent common ancestor between this group and this group? Well, you figure that out by just going back to the first place where these two things can converge on your phylogenetic tree. If you'd like, you can put your finger on each one and trace it back to where they run into each other. But the most recent common ancestor of these two is this point right here, right where it branches. Let's think about another one. Where is the most recent common ancestor between the Callinicrae and the Heliconiae? So here's Callinicrae, here's your Heliconiae. So where do these two tribes converge? Well, it's way back here. This is the first point that can lead to each of these. So like, why isn't it here? Well, then we would be excluding this upper group if you thought that it was here. Why isn't it right here? Well, this is not downstream of this lower group. We're going to talk more about this later, but hopefully this makes some sense. You have to understand how a phylogenetic tree works in order to be able to understand the evolutionary relationships necessary for this class. So, one major goal of studying zoology is to locate the origins of major characteristics. So when I say origins of major characteristics, I'm talking about where did these things evolve from? Because this is commonalities amongst many organisms. Not all animals have these things, but many of us do. So where did multicellularity come from? Where did coelom come from? Where did this idea of spiral cleavage, which we're going to get to later on, come from? I guess we're going to get to all these later on. Where do we get vertebrae from? Where did homeothermy originate from? And another goal is to understand the historical processes that generate and maintain diverse species and adaptations, which we will also discuss later. So where did these things come from, and why are they here, and what keeps them around? So we got to talk about the scientific process. Zoology is a scientific discipline. So let's talk about the essential characteristics. The first is that science is guided by natural law. It has to be guided by natural law. You cannot use supernatural explanations for things. It does not count. Okay. If you say that ghosts caused animals to evolve, well, you can't really get the ghost down to the lab, and so that would not count as science. So it must be guided by natural law, not conjecture, not guesses, not supernatural things, natural law. Science must be explanatory by reference to natural law. Again, you have to be able to go back to the underpinnings of natural law in order to be able to explain what you are saying. The conjectures of science are testable against the observable world, so science must be guided and explained by natural law, and it has to be testable. If you just say, like, I think, the sky is blue because I like blue, that doesn't count. It has to be testable. You can't test opinions. You can't test for things like ghosts. The conclusions of science are tentative and therefore not necessarily the final word. We don't prove things 100% in science. We are merely proving, we are merely discussing likelihoods and probabilities. Science is never settled. There are always things that can be proven or disproven. There's always more to be learned. Science is also falsifiable. So it has to be something that can be proven false. If it's based on opinions, if it's based on something that you have invented, it doesn't count as science. So the scientific method, or the hypothetic-deductive method as we often use it, is a scientific process of making a conjecture and then seeking empirical tests that potentially lead to its rejection. So in science, we always should be looking for something that proves us wrong. That is sort of the objective. We're testing null hypotheses versus alternative hypotheses. And one begins this process by generating a hypothesis or a potential testable explanation of some phenomena that we have observed in nature. So what is this hypothesis that we need for science? The hypothesis is a possible answer to a scientific question. It is based on prior observations or existing theories. And again, these are all conforming to these properties of science. Okay? Got to be natural law, has to be testable, has to be falsifiable. A hypothesis is a specific and testable statement that explains a narrow set of observations and can be supported or rejected through experimentation. So it would be a statement like, if we provide the plant more fertilizer, then it should grow taller. If we provide the plant with more light, then it will produce more fruit. Okay? It is a specific testable statement that explains a narrow set of observations and can be supported or rejected through experimentation. And it often takes the form of an if-then statement. If X, then Y. If we give the plant water, then it should grow larger. So continuing, the scientific method, there are different flavors of presenting the same information, but they all have the same general outline of steps. And the scientific method always starts with an observation. What's something that you see in nature? By the way, this is a Scottish philosopher, David Hume, who was really interested in the ideas of empiricism and that we aren't proving things just because two phenomena happen and appear to be related doesn't necessarily mean that they are. He was sort of one of the thinkers that helped us to develop the scientific method into what it is today. But the scientific method starts with an observation, something that you see in nature. And then it leads us to a question. Well, why did that thing happen? What led us to being able to observe this phenomena? And then we formulate a hypothesis, an if-then statement that should hopefully help us figure out what our question is that led from our observation. Next up, then, we have an empirical test. We have a controlled experiment using at least two groups, a test group and a control group, although it can consist of other types of groups as well. This is just a general outline. And then after we analyze our data, this leads us to conclusions, which allow us to accept or reject the hypothesis that we came up with. And we can publish our data that we were able to gather from this. The publication often leads us back to earlier parts of the scientific method. So if it was wrong, maybe you formulate another hypothesis, or maybe you can generate more observations, more questions from the experiment itself. So observation is a critical step in evaluating animal populations. For instance, there is this moth during the turn of the century called Byston bitularia, and you can see it has the melanistic form and then the more white form here. And what this chart is showing us is that as things, sort of the Industrial Revolution peaked before we started cleaning things up or desiring clean air and water, there was a huge amount of smoke. There was also a large number of melanic moths, the dark moths. But as we cleaned up the soot from the trees in England, the number of these dark moths declined drastically out of the 1980s into the 1990s. And so the question is, why do pigmentation patterns vary according to habitat? Why do we see so many dark moths in polluted areas when compared to these light moths? Or why do we see so many light moths in unpolluted areas? And so the hypothesis might be that coloration is influenced by a direct action of the environment. Maybe they're actually eating the soot. Maybe the soot is just getting on their wings and making them turn dark. So the null hypothesis would be that environment has no impact on coloration. Null hypotheses are generally simple explanations of things. So we are trying to disprove this. Okay, and so our empirical test, we take some moths, we take some light moths and some dark moths, and we breed them all in unpolluted environments. And if it's the soot itself that's making them turn dark, we should be able to see this. However, what we actually see is that moths retain the coloration of their parents, and this hypothesis is rejected. So it's not the soot that's making them dark. It's some other factor here. So another hypothesis is pigmentation is a genetic trait in Byston bitularia. Okay, and results show that dark pigmentation in the moth is genetically dominant, and we still haven't answered our question. What is it that makes dark moths live in polluted areas and light moths live in unpolluted areas? So this moth is active at night. It rests on the bark of trees during the day. And so the idea here is that moths that contrast with their background are preferentially selected against by predators that use vision to catch prey, such as birds, who are a major predator for these moths. So we place lightly colored and darkly colored moths in polluted and unpolluted branches, and they used clay models for this, and they went back and they saw that the dark coloration protects dark moths during the day. And so a dark moth that lands on a polluted branch is not going to get eaten as often during the day. And so this experiment led us to understand that this is a selective process taking place that's driving the differences between these two moths. I'm going to cut it off here. I think it's getting pretty long. All right. See you in the next one.